After you retrieve your plate from the incubator, you should notice that it has colonies on it. We will be using a single colony to grow a large-scale culture of E. coli. To begin with, we need to start a starter culture. Obtain a sterile 50 ml conical tube. Label it with the gene name, the plasmid name, the date, and your initials. Loosen the top of the tube. Obtain a 10 ml serological pipette. Fit it to an automatic pipetter or a manual pipetter, and loosen the top of the LB media. Although not shown in this video, you should be using sterile technique by using a flame. Carefully pipette 10 ml of LB media into the conical tube and repeat until you have 20 ml of LB in the conical tube. Add the appropriate antibiotics with the appropriate concentration to your LB. The type of antibiotic and the concentration of the antibiotic will depend upon the plasmid and the organism that you are growing. After you have set up your LB, we will be plucking a colony using a pipetter. Generally, a 20 microliter pipetter is useful. Obtain a sterile tip, carefully scrape the bacterial colony from the top of the plate. Make sure it's on the tip of the pipette. And eject the pipette tip directly into the LB. Place your inoculated culture in the incubator at 37 degrees with shaking. After the culture has shaken overnight at 37 degrees, the culture is saturated. Before we move on, we need to make sure that we collect cell stock. To make cell stock, we use an autoclaved 1.5 ml microcentrifuge tube. We pipette in 200 microliters of 80% glycerol. Ensure the glycerol is autoclaved and sterile. Next, we pipette in 800 microliters of our overnight culture. We make glycerol stocks so that we can use the stocks directly for the next time we need to grow an overnight culture. Ensure that you pipette up and down to mix the glycerol with the LB and cells. Use the remainder of the overnight culture to inoculate 1 liter of LB . Ensure that you add the appropriate amount of antibiotic for the plasmid and organism that you're growing. Place your inoculated culture in the incubator with shaking at 37 degrees. Occasionally, you will need to check the optical density of the cells at 600 nanometers. To do this, use a sterile serological pipette. Extract 1 to 2 milliliters of the culture and pipette it into a clean, disposable cuvette. To read the optical density, move to the spectrophotometer. Be sure to blank the spectrophotometer using water in the same style cuvette. Next, add your sample to the spectrophotometer and measure the reading at 600 nanometers. Once the optical density of your cell culture has reached between 0.6 and 1.0, we are ready to induce. Once the optical density of your culture has reached 0.6 to 1.0, we induce the cells with IPTG or arabinose, depending on the expression system. Add sterile IPTG or arabinose directly to the culture. Place the culture back into the incubator and drop the temperature to 20 degrees Celsius overnight. On the next day, retrieve your cells from the incubator. The solution should no longer be clear. Carefully pour your overnight culture into a 1-liter centrifuge tube. Balance the tube with water in another 1-liter centrifuge tube. Be sure the tubes are balanced. Place your tubes in a rotor on opposite ends. Carefully add the lid to the rotor. Tighten the lid down, but not too tight. Retrieve your samples from the centrifuge. Your cell pellet should be at the bottom of your tube. Pour your LB back into the flask that you retrieved it from. The cell pellet will stay towards the bottom of the tube. Next, add bleach to the spent media to approximately 10% so that the media becomes sterilized. When done with the media, we pour it down the sink and clean the bottle later. Tear a brand new 50 ml centrifuge tube. Label the tube with the protein name, the date, and your initials. Using a scoopula, carefully scrape the cell pellet from the tubes and scrape it into the 50 ml tube. Weigh the tube again and record the cell pellet weight on the front of the tube.